Here, we are going to talk today about the resolution of different physical mapping techniques. Resolution of different physical mapping techniques. Different physical mapping techniques. Under this, we'll just draw a tree wherein we classified the entire genome physical mapping techniques. The first one is restriction mapping. Restriction mapping. Under this, we clearly know that going beyond 50 kb length gets very difficult to use this particular technique for physical genome mapping. And the next technique that we have is optical mapping. The next technique that we have is optical mapping. Optical mapping. Under optical mapping, we have gel stretching, molecular combing, and the one that is currently in vogue is bio-nano. Bio-nano. We all know that these two techniques have been standardized with back clones having 1 MB in length. And the usage of this technique for lengths beyond 1 MB is limited. However, the resolution of this particular technique is 1 kb. Now, bio-nano, being a recent modification of optical mapping, stands out to be the best with having a resolution of 500 base pairs. When I talk about resolution, I mean that you can identify two markers clearly within a distance of this much. For example, when I say it is 500 base pairs, you can identify two markers, A and B, clearly within a distance of 500 base pairs using bio-nano. Now, we'll go to the next technique, that is fluorescent in situ hybridization. The next technique is what? Fluorescent in situ hybridization. Fish. Next technique is fish. Under fish, we have studied three variants. The first one is the metaphase fish. The metaphase fish, wherein the resolution is only 1 MB. And then we have studied a prophase fish or the interphase fish, prophase or interphase, wherein the resolution of this prophase and interphase fish is around 25 kb. Whereas you have the final version of the fish, that is the fiber fish. And the resolution of this fiber fish was found to vary between 1 to 400 kb. 1 to 400 kb. And finally, we have STS mapping, which I'll draw here as an extension. You have STS mapping. Under STS mapping, we have looked at two different variants. One is simple somatic cell hybridization. One is somatic cell. And the other one, which is a modification of somatic cell, is nothing but radiation hybrid mapping. Radiation hybrid mapping. Now, somatic cell hybridization, the technique here has got a resolution of 1 to 5 MB, whereas this radiation hybrid mapping has got a resolution of 100 kb. 100 kb. So this is regarding the comparison of the resolutions of all the different techniques that we have discussed. So you can have a screenshot of this particular comparison and keep it for yourself, which is very important as far as your exams are concerned. Thank you.